Hi everyone. Today we will discuss a very important topic in microbiology, the nutrition and growth of bacteria, and why these concepts matter across many fields. Understanding how bacteria obtain nutrients and grow is essential for microbiology, medicine, food safety, and biotechnology, influencing diagnosis, treatment, and industrial processes. Have you ever wondered how microscopic organisms can multiply rapidly and survive diverse environments? We will explain the mechanisms that enable survival and rapid reproduction. We will explore basic nutritional needs, gaseous and temperature requirements, and the processes and phases that define bacterial population growth over time. Let us begin by defining bacteria. Bacteria are prokaryotic, unicellular organisms with simple cellular organization, but complex metabolic capabilities that support diverse lifestyles. Unlike eukaryotic cells, bacteria lack a nuclear membrane and membrane-bound organelles such as mitochondria, Golgi apparatus, or endoplasmic reticulum, which affects their metabolism and structure. A typical bacterial cell includes a cell wall, cytoplasm, ribosomes, a nucleoid region, and sometimes flagella or pili used for movement and surface attachment. Cell walls vary widely. Gram-positive bacteria have thick peptidoglycan layers, while gram-negative bacteria possess thin peptidoglycan plus an outer membrane with lipopolysaccharides. Nutrients cross the cell envelope by passive diffusion, facilitated transport, active transport systems, and specialized channels such as porins in outer membranes. Macroelements required in large amounts include carbon, nitrogen, phosphorus, sulfur, oxygen, hydrogen, potassium, and sometimes calcium for structural and metabolic needs. Micronutrients or trace elements such as iron, magnesium, zinc, and copper function as cofactors for enzymes and are essential in small amounts for metabolic activity. Certain bacteria require external vitamins or growth factors like vitamin B derivatives because they cannot synthesize these compounds themselves and must obtain them from the environment. Bacteria are classified by energy and carbon sources. Phototrophs derive energy from light, while chemotrophs extract energy from chemical compounds, organic or inorganic. Carbon source determines autotrophy or heterotrophy. Autotrophs fix carbon dioxide into biomass, whereas heterotrophs rely on organic compounds for carbon and energy. Chemoheterotrophs use organic molecules for both energy and carbon, while chemolithotrophs oxidize inorganic substances like hydrogen sulfide or ammonia for energy production. Facultative bacteria can switch metabolic pathways depending on available nutrients and oxygen, allowing flexibility that supports survival in fluctuating environments. Understanding metabolic types helps predict growth requirements and design appropriate culture media for laboratory isolation, industrial fermentation, or biotechnological applications. Next, we examine gaseous and temperature requirements, key environmental factors that determine where different bacterial species can live and multiply. Oxygen requirements categorize bacteria into obligate aerobes, obligate anaerobes, facultative anaerobes, microorophiles, and aerotolerant organisms with distinct oxygen tolerances. Obligate aerobes require oxygen to grow and rely on aerobic respiration. Obligate anaerobes lack oxygen-dependent metabolism and are harmed or killed by oxygen exposure. Facultative anaerobes grow in the presence or absence of oxygen, usually preferring oxygen because aerobic respiration yields more energy per substrate molecule. Temperature preferences group bacteria as psychrophiles, mesophiles, thermophiles, or hyperthermophiles, each adapted to cold, moderate hot, or extreme heat ranges respectively. pH and water activity significantly influence growth. Many bacteria prefer neutral pH and high water availability, while acidotophiles and xerophiles tolerate different extremes. Salt tolerance varies among species. Halophiles actively thrive in high salt environments, whereas non-halophiles are inhibited by elevated osmotic pressures. In batch culture, bacterial populations follow 4 growth phases: lag, exponential, or log, stationary, and death, each reflecting physiological changes in the population. During the lag phase, cells adapt to new media, repairing damage and synthesizing enzymes and transporters required before starting active division and population increase. In exponential phase, cells divide at a constant rate determined by species and conditions, producing a steep, predictable upward slope on growth curves used to calculate generation times. Stationary phase occurs when nutrient depletion or waste accumulation limits growth, so that growth rate equals death rate and population size stabilizes temporarily. Death phase shows declining viable cell numbers. Some cells form spores or enter persistent states to survive adverse conditions long-term until conditions improve. Understanding nutrition and growth informs antibiotic strategies, optimizes fermentation and bioprocessing, and improves methods for controlling contamination and ensuring public health. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, Connecticut, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.